The Staphylococcus genus belongs to the Bacillus lactobacillus streptococcus cluster, which consists of the gram-positive bacteria with a low G-C contained in the chromosomal DNA. The second edition of Bargate's Manual of Systematic Bacteriology, updated in 2004, reclassified Staphylococcus genus in a new family named Staphylococcaceae. The Staphylococcaceae family is a part of the order Bacillus.
S. aureus is an opportunistic pathogen or commensal resident on host skin and mucosa in humans and animals. Staphylococci from carried sites can be spread and transmitted into the environment where they are able to survive for a long time. Staphylococcus aureus may act as a pathogen if it succeeds in entering the host by several mechanisms, such as skin trauma, inoculation, device implantation, both in immune-compromised patients and all those showing unaltered microbiota. In human beings, more than 80% of human-acquired S. aureus diseases are endogenous infections that are caused by strains carried in patients' noses.
Bacterial identification in the microbiology laboratory is traditionally performed by isolation of the microorganism and phenotypic analysis of its characteristics. Staphylococci are isolated from foods using selective and differential media to select for members of the genus Staphylococcus, such as Birdpack agar and mannitol salt agar. BP agar was developed by Birdpacker, and this formulation was first published in 1962. The presence of the salt and potassium telluride selects for the genus Staphylococcus, which reduces the telluride formation, forming black colonies. Birdpack agar is widely used and is included in many standard procedures for testing foods, cosmetics, or swimming pool water for the presence of Staphylococcus aureus. It may also be used for the isolation of Staph aureus from clinical specimens and is also called the ectolorite pyruvate agar. To purify Staphylococcus aureus, we prepared a culture medium by weighing 3.175 grams of Birdpacker into a diluent bottle and mixing it in 50 ml of distilled water, and the mixture was boiled to dissolve. The medium was then sterilized by autoclaving at 121 degrees Celsius for 15 minutes. The medium was cooled in a water bath at 47 degrees Celsius, after which 2.5 ml of egg yolk tellurite emulsion was added aseptically using a pipette. The medium was then mixed and placed in sterile petri dishes, just enough to cover the bottom of the dishes, and the dishes were covered and left to solidify. Using a permanent marker, quadrants were drawn on the petri dishes and labeled 1, 2, 3, and 4. A single colony of S. aureus was then inoculated and streaked on one of the petri dishes, with sterilizing the wire loop after streaking each section of the petri dish. We then incubated the medium for 48 hours at 37 degrees Celsius in order to isolate the microorganism of interest, and for this case, S. aureus. The isolated sample was then further purified by streaking on another petri dish and incubated for 48 hours at 37 degrees Celsius, after which purified S. aureus was obtained.
Birdpacker agar contains carbon and nitrogen, sources necessary for growth. Lithium chloride and potassium telluride act as selective agents by inhibiting the growth of accompanying microbial flora, whereas pyruvate and glycine selectively stimulate the growth of Staphylococci. The medium is differential by the formation of a diol allo, resulting from the production of lipase and lecithinase by coagulase positive Staphylococci strain, which breaks down lipids and lecithin in the egg yolk. Staphylococci produce dark gray to black colonies due to telluride reduction. An opaque zone of precipitation may form due to lipase activity. There is an ISO method available where BP agar is used for the enumeration of Staphylococci in food.
Coagulase positive Staphylococci, i.e. Staphylococcus aureus, produce dark gray to black, shiny convex colonies with entire margins and clear zones with or without opaque zones around the colonies. Coages negative staphylococci produce weak or no growth with grey or black colonies, usually without clear or opaque zones around them. Organisms other than Staphylococcus are often inhibited. If growth occurs, the colonies may be brown to grey to colorless. With neither clear nor opaque zones, the presumptive identification obtained on this media must be confirmed with additional tests.
An incubation of 46 to 48 hours is necessary for development of the typical appearance of S. aureus colony. Staphylococci, other than S. aureus, may grow on the medium. However, since their growth is dependent on the species and start by bucket agar must not be used for their isolation. Organisms other than Staphylococci may grow on this medium and may produce brown to black colonies. Example, Protas mirabilis. Therefore, additionally, tests are necessary for a complete identification of the isolate.
Typical colonies obtained in the media described above are submitted to gram staining to determine their purity and to observe their morphology and specific color. After confirmation of these characteristics, the isolates are submitted to catalase and coagulase tests. The tube coagulase test for the detection of free coagulase continues to be the gold standard for the identification of S. aureus and other coagulase positive staphylococci. The test should always be analyzed after 4 hours of incubation, since some strains will produce fibrinolysin when incubated for 24 hours. This causes dissolution of the clot during the period of incubation and may be erroneously identified as coagulase negative staphylococci if the test is read only after 24 hours. Although the cell wall of most S. aureus strains contains a bound coagulase called clamping factor that can react directly with fibrinogen in plasma mounted on a slide and produce rapid agglutination, the slide coagulase test is not recommended for differentiation from coagulase negative staphylococci because of some of these species such as S. lignensis and S. schleicheri subspecies. Schleicheri can also produce the clamping factor and may be erroneously identified as coagulase positive staphylococci.
Although the tube coagulase test is sufficient to identify S. aureus in human clinical samples because it's the only species present in humans that is able to clot plasma, the exclusive use of this test is not sufficient for the identification of S. aureus from foods of animal origin, where other coagulase positive staphylococci such as Staphylococcus hicas may be present. To overcome this issue, and to distinguish S. aureus from other species, testing of biochemical properties of strains such as fermentation of sugars, for example, trehalose, mannitol, xylose, and production of acetylene are suitable.
Until recently, microbiological methods dedicated to S. aureus identification were based on the use of selective growth media, which are time-consuming and preclude same-day diagnosis. The rapid and efficient identification of S. aureus is important for routine testing in microbiology laboratory. The development of reliable, simple, and inexpensive techniques is essential for identifying outbreaks and new strains of S. aureus. In recent years, different commercial systems for the identification of S. aureus have been developed as an alternative to classical identification methods. Automated systems and commercial kits based on miniature biochemical tests are widely used today, both in routine and research laboratories. However, these diagnostic systems have some disadvantages, such as their high cost. In addition, they are unable to reliably distinguish between different staphylococci species due to the variable expression of phenotypic characteristics, because they are based on colorimetric results, and subjectivity in their interpretation can lead to ambiguity. Molecular techniques have been the preferred method for identifying microorganisms because of their higher specificity and sensitivity. But the development of new methods for detecting S. aureus has been and will continue to be essential for our understanding of this organism and its relationship with food.
Like any other organism, S. aureus requires specific conditions for its optimum growth. Let's look at some of them. Although S. aureus grows well in culture media without sodium chloride, it can grow well in 7 to 10 concentrations, and some strains can grow in 20%. S. S. aureus has a high degree of tolerance to compounds such as tellurite, mercury chloride, neomycin, polymyxin, and sodium azide, all of which have been used as selective agents in culture media. Regarding pH, S. aureus can grow over the range 4 to 9.8, but its optimum is in the range 6 to 7. As is with the case with other growth parameters, the precise minimum growth pH is dependent on the degree to which other parameters are at optimum levels. In homemade mayonnaise, enterotoxins were produced when the initial pH was as low as 5.15 and when the final growth pH was not below 4.7. With respect to water activity, the Staphylococci are unique in being able to grow at values lower than any other non-halophilic bacteria. Growth has been demonstrated as low as 0.83 under ideal conditions, although 0.86 is the generally recognized minimum water activity. Staphylococci are typical of other gram-positive bacteria in having a requirement for certain organic compounds in their nutrition. Amino acids are required as nitrogen sources, thiamine and nicotinic acid are required among the B vitamins, and when grown anaerobically, they appear to require uracil. In one minimum medium for aerobic growth and enterotoxin production, monosodium glutamate serves as carbon, nitrogen, and energy sources. This medium contains only 3 amino acids, namely arginine, cysteine, and phenylalanine, and 4 vitamins known as pantothenic acid, biotin, niacin, and thiamine. In addition to inorganic salts, arginine appears to be essential for enterotoxin B production. Although it is a mesophile, some strains of S. aureus can grow as low as 6.7 degrees Celsius. In general, growth occurs over the range 7 to 47.8 degrees Celsius, and enterotoxins are produced between 10 to 46 degrees Celsius, with the optimum being between 40 and 45 degrees Celsius. These minimum and maximum temperatures of growth and toxin production assume optimal conditions relative to other parameters such as temperature, pH, water activity, and oxidation reduction potential. S. aureus cells are considerably more sensitive to heat than the enterotoxins. In general, enterotoxin production tends to be favored by the optimum growth conditions of pH, temperature, and oxidation reduction potential. It is well established that Staphylococci can grow under conditions that do not favor enterotoxin production. With respect to water activity, enterotoxin production except for Staphylococcal enterotoxin A occurs over a slightly narrower range than growth.
S. aureus is an opportunistic microorganism that is prominently known for food poisoning. Here's an insight of some of the study cases. The staphylococcal food poisoning or food intoxication syndrome was first studied in 1894 by J. Dennis and later in 1914 by M.A. Baba, who produced in himself signs and symptoms of the disease by consuming milk that had been contaminated with a culture of Staphylococcus aureus. The capacity of some strains of S. aureus to produce food poisoning was proved conclusively in 1930 by Duck, who showed that the symptoms could be produced by feeding culture filtrates of S. aureus. Although some authors refer to food-associated illness of this type as food intoxication rather than food poisoning, the designation gastroenteritis obviates the need to indicate whether the illness is an intoxication or an infection. Staphylococcal gastroenteritis is caused by the ingestion of food that contains one or more enterotoxins, which are produced only by some Staphylococcal species and strains. Enterotoxin production is believed to be generally associated with S. aureus strains that produce coagulase and thermonuclease. Staphylococcal gastroenteritis is considered to be one of the most frequently occurring foodborne diseases worldwide. The presence of S. aureus in foods potentially represents a public health hazard because many strains produce enterotoxins. Toxins are produced when the food, primarily protein-rich food, is left at room temperature for long periods. Staphylococcal enterotoxins are heat-stable proteins of molecular weight 26 to 30 kilodaltons and differ in toxicity. They are also water-soluble and may be resistant to proteolytic enzymes. Although staphylococcal enterotoxins form a group of serologically different proteins, namely A, B, C1, C2, C3, D, F, G, H, I, J, K, L, M, and O, they all share important features as they have similar structures. They all cause emesis and gastroenteritis They show super-antigenicity properties and are resistant to heat and pepsin digestion.
The most common presentation of Staphylococcus food poisoning includes nausea, vomiting, abdominal pain, diarrhea, chills with or without fever, dizziness, and others. Symptoms develop 2 to 4 hours after the ingestion of toxin-contaminated products, and the illness may vary from quite mild pictures lasting a few hours only and usually self-limiting to severe conditions, even fatal, that require hospitalization, mostly among children and the elderly. The exact mechanism on which Staphylococcus food poisoning relies is not yet completely understood. Proof of Staphylococcus food poisoning is established by recovering enterotoxigenic staphylococci from leftover food and from the stool cultures of victims. Attempts should be made to extract enterotoxin from suspect foods, especially when the number of recoverable viable cells is low. The minimum quantity of enterotoxin needed to cause illness in humans is about 20 micrograms. Nevertheless, it is known that meat, particularly if minced, salads, cream-filled bakery products, and dairy products are the main vehicles of enterotoxins, S. aureus isolates.
Most domesticated animals harbor S. aureus. Staphylococcal mastitis is not unknown among dairy herds, and if milk from infected cows is consumed or used for cheese making, the chances of contracting food intoxication are excellent. There is little doubt that many strains of this organism that cause bovine mastitis are of human origin. However, some are designated as animal strains. In one study, staphylococcal strains isolated from parts of raw pork products were essentially all of the animal strain type. However, during the manufacture of pickled pork products, these animal strains were gradually replaced by human strains during the production process, to a point where none of the original animal strains could be detected in finished products.
In general, staphylococci may be expected to exist at least in known numbers in any or all food products that are of animal origin, or in those that are handled directly by humans, unless heat processing steps are applied to effect their destruction. They have been found in a large number of commercial products by very many investigators.
In general, the staphylococci do not compete well with the normal biota of most foods, and this is especially true for those that contain large numbers of lactic acid bacteria, where conditions permit the growth of later organisms. Many investigations have shown the inability of S. aureus to compete in both fresh and frozen foods. Staphylococcus enterotoxin A has been shown to be resistant to a variety of environmental stresses, but growth of several lactic acid bacteria did lead to its reduction and to a suggestion that toxin reduction might have resulted from specific enzymes or other metabolites of the lactic acid bacteria. For example, Lactobacillus gasseri, when employed in a meat fermentation, was shown to prevent enterotoxin formation by Staphylococcus aureus in a model sausage preparation.
Glucose has been reported to decrease heat protection for S. aureus. Non-metabolizable sugars and polyols such as arabinose, xylose, and sorbitol have been found to protect S. aureus against sublethal heat injury, but the mechanism of this action is unclear. The sorbates are primarily effective against molds and yeast, but research has shown its effectiveness against Staph aureus as well. The combination of sorbate plus reduced nitrite is effective against S. aureus at high concentrations, and the effectiveness increases as the pH is lowered.
When susceptible foods are produced with low numbers of staphylococci, they will remain free of enterotoxins and other food poisoning hazards if kept either at or below 40 degrees Fahrenheit or above 140 degrees Fahrenheit until consumed. For the years 1961 to 1972, over 700 foodborne disease outbreaks were investigated by Bryan relative to the factors that contributed to the outbreaks. And of the 16 factors identified, the 5 most frequent factors involved were inadequate refrigeration, preparation of foods far in advance of planned service, infected persons practicing poor personal hygiene, inadequate cooking or heat processing, and holding food in warming devices at bacterial growth temperatures. Inadequate refrigeration alone comprised 25.5% of the contributing factors. The 5 listed contributed to 68% of the outbreaks. However, in recent years, the number of staphylococcal food poisoning outbreaks has declined. This decline is probably a reflection of the better use of refrigerated temperatures to store food and improve sanitary practices that can control contamination and growth of Staph aureus. Thank you.
Well, this has been our discussion about Staphylococcus aureus. Feel free to like our video.
And don't forget to hit the subscription button for more content coming from the Department of Food Technology and Nutrition here at Makerere University.
The hill of endless opportunities.
We build for the future.